Welcome to Biotanemy. Digestion and absorption of starch. Foods such as rice, wheat, potatoes, and bread contain starch, a polysaccharide composed of glucose units, in the form of amylose and amylopectin. Digestion begins in the mouth by the enzymes salivary amylase or ptyalin, which will break starch into maltose, maltotriose, and dextrins. As the food reaches the stomach, acidic pH inactivates salivary amylase. So, very little carbohydrate digestion occurs, and starch digestion temporarily stops. Swallowing food without proper chewing leads to larger food boluses and reduced mechanical breakdown in the oral cavity. This can increase gastric workload and delay gastric emptying, resulting in post-meal heaviness and prolonged satiety. In addition, swallowed air or aerophagia and delayed digestion may contribute to gastric distension, leading to burping and abdominal discomfort. In some cases, delayed carbohydrate digestion can increase substrate availability for colonic microbiota, leading to bacterial fermentation that produces gases such as CO2, H2, and CH4, resulting in flatulence and sometimes foul-smelling gas. Increased intragastric pressure may also predispose susceptible individuals to acid reflux and belching. Hence, remember to chew the food properly before you swallow to break down the carbohydrates as much as possible. The digestion continues in the small intestine. In the duodenum, the pancreas releases pancreatic amylase, which further breaks starch into maltose, maltotriose, and alpha-limit dextrins. The brush border enzymes on the intestinal microvilli, such as maltase, isomaltase, sucrase, and lactase, complete the digestion, resulting in starch breaking down into glucose, fructose, and galactose. In the jejunum, the glucose and galactose will be absorbed into the enterocyte along with sodium by sodium-dependent transporter. This is a secondary active transport. This sodium gradient is maintained with the help of sodium-potassium ADPase, located on the basolateral membrane, to pump out sodium inside the cells at the expense of 1 ATP to create low intracellular sodium ions, allowing SGLT1 to function. While the fructose is taken up into enterocytes by glucose transporter 5 through insulin-dependent facilitated diffusion. Inside cells, galactose is converted to glucose through the Lewar pathway. In this pathway, the galactose is phosphorylated by the enzyme galactokinase to galactose 1 phosphate with the expense of 1 ATP. Then the enzyme galactose 1 phosphate uridyl transferase, the major enzyme associated with classic galactosemia, will add uridine diphosphate or UDP from UDP glucose to galactose to form UDP galactose and glucose 1 phosphate. The UDP galactose produced in this step is converted back to UDP glucose by UDP galactose 4 epimerase. And glucose 1 phosphate will be converted to glucose 6 phosphate by phosphoglucomutase and enter glycolysis. On the basolateral side, the glucose transporter 2 or GLUT2 will move glucose into the blood by facilitated diffusion. In the blood, the glucose enters the hepatic portal vein and liver and is distributed through systemic circulation. Different tissues use different glucose transporters to take in glucose. The brain uses GLUT3, the liver uses GLUT2, muscle and fat use GLUT4, and RBC uses GLUT1. Fructose, on the other hand, is partially metabolized in enterocytes, leaves the enterocyte through GLUT2 to the portal vein to reach the liver. Inside hepatocytes, it will join glycolysis with the help of fructokinase, converting fructose to fructose 1 phosphate at the expense of 1 ATP. Glucose crosses the plasma membrane through these GLUT transporters and enters the cytoplasm, and the process of glycolysis begins to produce energy. Glycolysis is a central metabolic pathway consists of 10 steps to occurs in the cytoplasm and functions under both aerobic and anaerobic conditions, in which one molecule of glucose is converted into 2 molecules of pyruvate through a series of enzyme-catalyzed reactions, producing ATP and NADH. The 10 enzymatic steps are divided into 2 phases. Phase 1, from step 1 to 5, is called the energy investment phase, which uses ADP to activate glucose. Phase 2, from 6 to 10, called the energy payoff phase, produces ATP and NADH. Let's see the reactions in the energy investment phase. Step 1. Phosphorylation of glucose. The enzyme hexokinase is found in most tissues, and glucokinase in the liver and pancreas will convert glucose to glucose 6-phosphate by taking one phosphate from ADP to convert it into ADP. This step is an irreversible reaction and traps glucose inside the cell. Step 2. Isomerization. The enzyme phosphoglucose isomerase will isomerize glucose 6-phosphate to fructose 6-phosphate, or it'll convert aldose sugar into ketose sugar. Step 3. Second phosphorylation. This is the second irreversible step, phosphorylating fructose 6-phosphate to fructose 1,6-bisphosphate using the phosphate group from ADP, converting ADP to ADP, catalyzed by phosphofructokinase 1. The second irreversible step of the reaction. It is a rate-limiting step that is activated by AMP, ADP, and fructose 2,6-bisphosphate and inhibited by ATP and citrate. Step 4. Cleavage reaction. Here, the enzyme aldolase will split the 6-carbon molecule into two 3-carbon molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Step 5. Interconversion of triose phosphates. It is the final step of the energy investment phase. Here, the enzyme triose phosphate isomerase will convert dihydroxyacetone phosphate to glyceraldehyde 3-phosphate, which means 2-glyceraldehyde 3-phosphate. Next, let's look into the energy payoff phase. Step 6. Oxidation and NADH formation. It is an oxidation step. The enzyme glyceraldehyde 3-phosphate dehydrogenase will convert glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate and NADH. Step 7. First ATP generation. With the help of enzyme phosphoglycerate kinase 3-phosphoglycerate to produced from 1,3-bisphosphoglycerate by releasing 1-phosphate from it to ADP to produce 1 ATP. Step 8. Rearrangement. The enzyme phosphoglycerate mutase will convert 3-phosphoglycerate to 2-phosphoglycerate. Step 9. Formation of phosphoenol pyruvate. In this step, the enzyme enolase 2-phosphoglycerate will dehydrate to phosphoenol pyruvate. The presence of fluoride can inhibit this reaction. Step 10. Formation of pyruvate. This is the third irreversible step that produces ATP with the help of the enzyme pyruvate kinase and pyruvate from phosphoenol pyruvate. Fructose 1,6-bisphosphate is an activator of this step, while it is inhibited by the presence of ADP and alanine. The high-energy molecules produced during glycolysis are transported to the electron transport chain. So, the intermediates can be remembered by... Goodness gracious, Father Franklin did go by picking pumpkins to prepare pies. Overall glycolysis yield is 2-pyruvate for ADP, 2 NADH, 2 hydrogen ions, and 2 water. Hence, the ADP used in these reactions are 2, while produced is 4. NADH produced is 2. The net energy yield of this cycle during aerobic conditions is calculated as 2 ADP plus 2 by 2.5 ATP equals 7 ADP. NADH itself cannot cross the mitochondrial membrane for ETC, so they use two shuttle systems, the malate, aspartate shuttle, and the glycerophosphate shuttle. Malate, aspartate shuttle transfers NADH to complex 1 in the liver, heart, and kidneys. The cytosolic NADH gives electrons to oxaloacetate to form malate and NAD+, with the help of the enzyme malate dehydrogenase. Malate can cross the inner mitochondrial membrane through malate, alpha-ketoglutarate antiporter. Inside the mitochondrial matrix, malate will be converted back to oxaloacetate by mitochondrial malate dehydrogenase to produce mitochondrial NADH. The NADH produced now joins the ETC through complex 1. And in brain and skeletal muscle, the glycerol 3-phosphate shuttle or glycerophosphate shuttle will occur. Here, the cytosolic NADH produced during glycolysis will donate electrons to dihydroxyacetone phosphate, regenerating NAD+. Then the mitochondrial glycerol 3. Phosphate dehydrogenase transfers those electrons to FAD to form mitochondrial FADH2 and enters the ETC at complex 2. In anaerobic conditions, without oxygen, the electrons cannot move through the ETC and NADH, and FADH2 cannot unload electrons. ADP production by oxidative phosphorylation stops. So, the enzyme lactate dehydrogenase converts pyruvate to lactate at the expense of 1-NADH. So, the net energy yield is 2 ADP only. This reaction will regenerate ND for continued glycolysis in anaerobic conditions.
Pyruvate inside the mitochondrial matrix will be converted to acetyl-CoA by the pyruvate dehydrogenase complex.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.